Human cells became something of a flashpoint in the past year, cited by some as the reason they're hesitant to get the COVID-19 vaccine. With us now for the science and to clear up some of the misinformation out there, from High Park in the provincial capital, there's Dr. Janet Rossant. She is senior scientist at the Hospital for Sick Children and president of the Gairdner Foundation. Dr. Rossant, it's good to have you on TVO tonight. How are you doing?
I'm doing fine. It's great to be here.
Great to hear. Some people apparently are hesitant or will not take the COVID-19 vaccine because they believe that the vaccine contains fetal cells from abortions. So let's get that straight once and for all. Is that true or false?
That is false. There are no fetal cells in any of the COVID vaccines.
So if you don't want to take the vaccine and you're using that as the reason why you don't want to take it, you're all wet on this one. Is that right?
You are all wet if you think it contains fetal cells. It is true that some of the research behind the vaccines used cell lines derived from elective terminations of pregnancy. Cell lines generated many years ago that turn out to be very good at producing proteins and viruses that are needed to help develop vaccines. But there are no fetal cells in the vaccines themselves.
Okay, can you pursue that a bit for us? How are cells from aborted fetuses used in labs now?
So the fetal cell lines that were derived many, many years ago have been grown in culture for many generations. They divide indefinitely. They are nothing like the fetal cells that they began with. But they're, as I say, they're very good, they're very stable, and they produce some of the viruses and the vectors that are used to make vaccines. The messenger RNA vaccines that we know most about, the Moderna and the Pfizer-BioNTech ones, the fetal cell lines were used a little bit in the development, but when you produce the vaccines, they are made out of nucleic acid, and they don't have anything to do with fetal cell lines. The AstraZeneca cell line does use a cell line that was originally a fetal cell to produce the vaccine. But again, no fetal tissue is actually in the vaccine itself.
And are fetal cell lines ever injected into people?
No. These are research tools, technology tools to produce vaccines. In fact, they have been used for many of the vaccines that we're familiar with today that are saving lives. Rubella vaccines, rabies vaccines, some of the chickenpox vaccines, hepatitis A, all were using these cell lines that were originally, many years ago, derived from fetal material.
All right, let's talk about another kind of stem cell, embryonic stem cells. First of all, what are they?
Well, they also don't appear in vaccines, for sure. But embryonic stem cells are quite a different story. They are cell lines that are derived from early human embryos at the time when the embryo has about 100 cells. So it's the size of a speck of dust. And at that stage of development, the embryo hasn't made any decisions about what it's going to do. So the cells are what we call pluripotent. It contains these cells that can make every cell type in your body, because that's how we get from an egg to a person. And it turns out, first in the mouse and then in the human, that people were able to take these embryos at that stage and generate permanent pluripotent stem cell lines. And they are derived in humans from embryos that come from in vitro fertilization clinics, embryos that would not be used for reproductive purposes and have been donated with permission for research purposes.
So do you get all of yours from IVF clinics, for example?
Well, so most of my work is mostly with mouse embryonic stem cells, but in fact, the stem cell lines, there are human stem cell lines that have been approved in Canada, and we do some research with those. And yes, they were all, and there aren't a lot of them, there are few cell lines that are used quite a lot, and they were derived from in vitro fertilization clinics, yes.
And let me just clarify again, and why would you use this particular type of stem cell line?
Well, these are the cells that have incredible potential for regenerative medicine. Because they can grow forever in a petri dish, you have an endless supply of cells that can make nerve cells, that can make muscle cells, they make heart cells, they make pancreatic islet cells that produce insulin, they make the cells in the brain that make dopamine. So these cell lines can be used to generate cells that can help treat serious degenerative diseases.
So they're extremely useful for your purposes.
Yes, well, my purpose is hopefully for many, many people all over the world. There's a huge amount of activity in this area. But of course, it is controversial. They come from embryos. And so there's another kind of cell line that has many of the same properties. It's called an induced pluripotent stem cell. And they were produced first by a Japanese scientist, Shinya Yamanaka, in 2006, first in the mouse and then in the human. And he showed that you could take a skin cell and add back some of the genes that we and others had shown were important for the early embryo, add them back to these cells and essentially turn back the clock. And then these cell lines behave like embryonic stem cells. So now you don't have to use an embryo. You can get the same kind of potential in these cells by just taking adult skin cells. So this has really been a big boost to study these cell lines and also to study disease, because it means that if you have a genetic disease, you can make induced pluripotent stem cells and study that disease in a petri dish and develop new therapies. So these are incredibly powerful cells.
Okay, so you got to say their name one more time. I always call them IPS because that acronym is easier than the whole thing. But what are they actually fully called?
Induced, because they've been induced by adding these genes. Pluripotent, because they have many, many potentials. And embryonic stem cells.
Okay, let's do a little comparing and contrasting here, because the first type of cells that we talked about off the very top, you said come from generations ago. The embryonic stem cells, are they also from generations ago?
Well, they are now. So some of the embryonic stem cells that are being used quite extensively worldwide, there's really only a few cell lines that have been stable enough to be able to be passaged for many, many generations. And we're now looking at the first human embryonic stem cells were derived in 1998. So it is quite a long time, and they have been grown not continuously all that way, but they've certainly been through many generations in the petri dish. They just keep dividing.
And IPS stem cells, are they the same as well?
Yeah, they can divide indefinitely as well. But they were later, so they're 2007, so they haven't been around quite as long.
Gotcha. And have they come to replace embryonic stem cells in your lab use?
It's a mixed bag. There's no question that the IPS cells come with less concerns about their origins, and they do, in some cases, have special properties because you can derive them from humans with different genetic backgrounds. So there's a lot of use of IPS cells, but embryonic stem cells are still used in many labs because they're the sort of gold standard. We know where they came from. They came from the stage where we know what the cell behavior is, whereas the IPS cells, you've artificially made them. And so it's still a little bit, there are many, many IPS cell lines, but trying to work out which are the best ones to take forward for treating patients is still embryonic, I would say.
All right, one more question on this line, and that is, I suspect there are many people watching this right now or affiliated with this issue who have been asked to store the umbilical cord blood when they have a baby. So tell us about how that is used.
Yes, so from the umbilical cord, you can isolate stem cells, but they are not embryonic stem cells. They are not cells that can make every cell type. They are actually the stem cells that make your blood. So they are like bone marrow stem cells. They can be used for bone marrow transplantation. But really and truly, what we need to do, the best way to store umbilical cord blood is to store it in a public cord blood bank, the same way we store blood and we store other kinds of cells. Because if you put your baby's cord blood into a public cord blood bank, that can be pulled by anyone who needs it, a child who needs it later on. And you can go back in and pull out cells for yourself. But a single cord blood is, generally speaking, not enough to treat one person. So a private bank that says, I'll store it just for my child, is not the best way to go.
You prefer the public situation?
Absolutely.
Yeah. And is it the same thing if you're, for example, getting a blood transfusion or you need blood? There's a match that has to happen or it's no good. Is that the same thing here?
Yes, yeah, you have to, absolutely, it has to be matched.
Gotcha. All right, we've talked about all these different kinds of stem cell lines. Let's talk about how they're employed. And that is, how many of the treatments or products do we use which employ these cells in their research and development?
So we're talking about fetal cells or embryonic stem
Yes,
cells?
yes. Well, okay, you tell us. I mean, both, I guess.
Well, as I say, fetal cell lines are used still quite extensively for research purposes and to some degree for producing vaccines. Embryonic stem cells and iPS cells are moving now towards clinical trials. We are seeing the first clinical trials for using embryonic stem cells, or products of embryonic stem cells, I should say, for treatment of diabetes, Parkinson's, macular degeneration. Those are ongoing clinical trials. The diabetes one, there's quite a lot of researchers involved in that in Canada, for example. Parkinson's in Japan and in Europe, and macular degeneration, also Japan, US, and Europe, from taking embryonic stem cells in the petri dish, differentiating them into the right cell types and putting them back in patients. So these are early phase clinical trials, but some of the data is really looking quite promising.
Let's understand what that means. Early phase, quite promising, meaning we're still probably many years away from touchdown?
Well, this is always, of course, a difficult question, I'm sure you're aware. The early phase trials are really trying to say, are these things safe, and do we see any efficaciousness? And the answer is for some of these, that seems to be the case. I think some of the ones like macular degeneration, when they're actually replacing the pigmented cell in the eye which degenerates, look as though if those work, it could be relatively quickly turned into a clinical use as opposed to just a clinical trial. So we're looking at, you want me to guess? I mean, I do think we'll be seeing much broader clinical use of some of these approaches within 5 years.
But if I were to put you on the spot right now and say, what is the one thing that you hope all of this specifically will lead to as it relates to disease X, Y, or Z, what would you say?
Well, I think that the power of embryonic stem cells is that we can produce cell types potentially to treat diseases for which we really don't have a curative type of treatment. Take type 1 diabetes. Patients are on insulin for a very, all their life, and there are many complications that go along with that. We know that you can transplant pancreatic islets from donor people who die into patients and get them off insulin for years. But we don't have enough pancreatic islets. Embryonic stem cells give you the potential to make those pancreatic islet cells in large quantities and potentially really treat large numbers of patients with type 1 diabetes. So that's the sort of promise that is out there and is coming down the road.
Are you specifically working on that in your lab?
No, I'm a stem cell biologist, but I'm really interested in just what a stem cell is and how they come from an embryo and what are the pathways that make a pluripotent cell special. And in my lab, in the mouse, we derive stem cells from that same early stage of development, but we derive stem cells that make the placenta, because pluripotent embryonic stem cells actually don't make the placenta. So we want to understand how those two cell types develop in the embryo. And actually what we'd like to do is make what we call a totipotent cell line, a cell line that could make everything, including the placenta, so that we could really model and study early embryonic development in the petri dish and the placenta and find out what goes wrong in a lot of pregnancy disorders as well.
You know, I don't know this for sure, but I'm betting you love your job.
I do. I think scientists have a passion. If you don't have a passion for what you're doing, why are you doing it? And I've worked on early embryonic development for all my career, and my lab is getting smaller and smaller these days. But the fact is that all the work that I've done over the years, other people are now still doing, and there are lots of new tools that study how embryos develop. And all the things we have said about stem cells could be used to help treat people are beginning to come to pass. So that's very satisfying.
That you love your job, it really shows. Dr. Rossant, it's awfully good of you to spend some time with us on TVO tonight. Dr. Janet Rossant, Hospital for Sick Children, Gairdner Foundation, all good wishes going forward.
Thank you very much, Steve.
The Agenda with Steve Paikin is made possible through generous philanthropic contributions from viewers like you. Thank you for supporting TVO's journalism.